Okay, so let's just keep going with our chemistry review. So remember that we have inorganic and organic molecules. Organic molecules are going to contain carbon and hydrogen, and those are the ones that we're going to see the most often when we eat our food. So carbohydrates, lipids, proteins, nucleic acids, and then some are going to be our acids and bases. And then in the inorganic side, we have water and electrolytes. Electrolytes is a fancy word for ions, and if we remember what an ion is, that is a positive or negatively charged atom. So the ions that we talk about most often are going to be potassium. Remember that K plus, chloride, which is the Cl minus, sodium, which is an Na, and then a plus sign. Okay, and then some of those acids and bases. So pH is really relative to this, and it helps us to maintain homeostasis. So remember, homeostasis is maintaining that balance. So pH is a measurement of the hydrogen ions in a solution. So when we put the solution into water, what happens? How does it dissociate? So we get those hydrogen ions, or we can get hydroxyl ions. So the pH scale is going to range from 0 to 14. Pure water is about 7. We have less than 7 is going to become acidic, and then higher than 7 is basic. We also call that alkaline. The pH of human blood is going to be on average about a 7.4, but it can range from about 7.35 to 7.45. Okay. So we have these carbohydrates. Now remember, carbohydrates are carbon. That's where that carb comes from. O is the oxygen, and hydrates is that water part, right? So it hydrates us like water. So carbohydrates is made out of carbon, oxygen, and hydrogen. It has rings of carbon attached to hydroxyl groups. There are saccharides, so we have monosaccharides. Monosaccharides are great for energy, so glucose and fructose. We also have disaccharides, which are sucrose and lactose. We have polysaccharides. This is how we store those saccharides. So we can have glycogen. Animals are going to store through glycogen, but plants are going to store through cellulose or starch. And you can see that all that is if you look at the picture of glycogen there, that it is that glucose plus glucose plus glucose plus glucose plus glucose is going to equal glycogen. So some important concepts here is that humans are only going to burn glucose for food. That was a fact. Now it's maybe not a fact. And so we're not quite sure if that's truly the case, but we tend to burn glucose. In an ideal situation, we would love to use glucose, but our body can actually burn other things. Glycogen is stored in the liver and in our skeletal muscles, and we do what's called glycogenesis. So remember that word genesis means to create. So we can create glycogen from glucose. We also can do glycogenolysis. Remember that lysis part means to break it apart. And so glycogenolysis means to break the glycogen down into glucose. And then we have gluconeogenesis. That means to make new glucose. And when we make new glucose, we do it from using amino acids and glycerol from lipids. So lipids, again, those are basically fats. Okay, so we have monoglycerides. Those are great for digestion and lung function. Diglycerides are most abundant in the body, and they help to build our body structures. And triglycerides is our primary storage of fat. Fatty acids can be both saturated or unsaturated. Unsaturated can be omega-3 or omega-5 fatty acids. And fatty acids and glycerol are the preferred fuel source for many of our tissues. Proteins are the most abundant and important organic molecules. I cannot say that enough. Proteins are the most abundant and important organic molecules for our bodies. Amino acids, we have about 20 of them in our body, and they are nitrogen containing compounds. They are sort of like pearls on a necklace, so to speak. And what they do is that we get this sort of chain of amino acid, and then they're going to start to fold over onto themselves and form some hydrogen bonds between them. We get what's called a secondary structure, and then it's going to fold onto itself and form a tertiary structure. And we take a whole bunch of them and stick them together, we get the quaternary structure, and that's going to be that functional protein. Again, it's formed by just about 20 different amino acid combinations, and we have both essential and non-essential amino acids. So essential means that we need to get it from our diet. Non-essential means that we do not. So proteins, remember, they can act as enzymes, and enzymes are catalysts. They make the reactions go faster by lowering the activation energy. So you can see that from this graph here, the pink line or the red line, it has lowered that activation energy required. And so when we can lower the energy required, that means that we can make the reaction go a little bit faster. Proteins are not changed or used up within that reaction. Those enzymes are not. They are specific because they only work on limited types of substrates. They're almost like a puzzle and they fit just perfectly. They are limited by their saturation, which means that you have so many enzymes, and once those enzymes are saturated, we can't do any more, right? So as many as we have, we're kind of limited by that. And they are regulated by other cellular chemicals. Nucleic acids would be like our genetic material, so our DNA or deoxyribonucleic acid, as well as ribonucleic acid or RNA. Okay. Nucleotides have a nitrogen base, a pentose sugar, and a phosphate functional group, and they are very large organic molecules. We find them within the nucleus, and they can store and process information at the molecular level. They also can store energy. ATP, for instance, is a nucleic acid. And we have different types of RNA. We have messenger RNA, transfer RNA, and ribosomal RNA. And as far as DNA goes, remember we have different sort of bases on there. We have adenine, thymine, cytosine, and guanine. And then for our RNA, we have adenine, uracil, cytosine, and guanine. So ATP, again, is that nucleotide, and it can be used to store energy. We also have ADP, adenosine diphosphate, rather than adenosine triphosphate. Tri means 3, di means 2, so it has to do with the number of phosphate groups on there. Okay? So we have what's called ATPase. Remember, ASE at the end is going to tell us that it is an enzyme, and ATPase is an enzyme that catalyzes the phosphorylation. So that means that it's going to add an extra high energy phosphate group to the molecule. Okay, so when we sort of add that in, we're actually storing that phosphate molecule. We're storing the energy within the phosphate bond. So let's talk a little bit about cells really quickly. Cells are surrounded by a water medium called extracellular fluid. That's going to be outside of the cell. The plasma membrane is going to separate the cytoplasm from that extracellular fluid, and the cytoplasm or the cytosol is the liquid that contains the organelles. Okay? And so remember, on the outside of the cell, we have that double-layered phospholipid membrane. You might have heard it called the phospholipid bilayer. Okay, phospholipid, what does that mean? It means that it's made out of fats and phosphate groups, right? And so since we have sort of that double layer, you can see it here. We have this double layer bilayer, I guess is what you could call it, this lipid bilayer. We have these phospholipids, steroids, and proteins, and you can see that you have this sort of head to it and then the tail. And so we have these hydrophobic tails. Hydrophobic means that it's sort of afraid of water. Or it doesn't like water. And so what happens is that they orient towards one another, and then the heads are hydrophilic, and so they orient outside so that that's towards the watery environments of the extracellular and the intracellular fluid. Okay. So that cytosol has some dissolved materials in it, like nutrients, ions, proteins, and waste products. It has very high potassium and low sodium. Also has some high protein, high carbohydrates, and low amino acid and fat. And then we have these organelles in there, and they have these structures in them with some specific functions. Okay, so make sure that you do go over these. I'm not going to read them all to you, but do review the organelles and their functions. Some specific ones would be the mitochondria. Make sure that you review that. Its job is to sort of transform and produce 95% of the ATP that's required for the cell. It's sort of the energy transformer of the cell. We have the rough ER and the smooth ER. Those are the endoplasmic reticulums, which is its big job is with protein synthesis and packaging. We have the Golgi apparatus, the lysosomes, the peroxisomes, and then here is the nucleus covered in the nuclear envelope, and then it has the nucleolus. Okay. So that mitochondria, again, that's the energy transformer of the cell, and it's going to perform aerobic metabolism. And so that's essentially cellular respiration. What is cellular respiration? This is the equation right here for cellular respiration. C6H12O6. What is that? That's glucose. Glucose in the presence of oxygen is going to yield water carbon dioxide, and ATP. So the big thing is that ATP, the carbon dioxide, we actually exhale it back out with our lungs, right? And our body sort of uses water as that waste product. Here's the nucleus. It's considered the control center of the cell. It's going to store all of that genetic instructions for manufacturing the proteins. So remember, the proteins are the most abundant, right? Most abundant and the ones that we use the most, okay? So that has the genetic code inside. Remember, we have a triplet code. So 3 bases is going to equal to 1 amino acid. So we might have, I don't know, AT or AUG, and that's going to code for one of those amino acids. It's also, remember, we have multinucleated muscle cells, and red blood cells don't contain a nucleus. So give it a second and just sort of compare those cell organelles to a school. So here we go. When we compare them to a school, we have the front entrance of the school. Okay, so that would be like the cell membrane. It's going to regulate what enters and leaves the cell. The main office, like the nucleus, it controls all the cell's activity. This is where the papers are coming in and out, determines what proteins are going to be made and the location of the DNA. The halls of the school would be like the cytoplasm, contains all those organelles and the site of the most cell activities. The kitchen of the school is going to be like the endoplasmic reticulum. It's where the ribosomes do their work, and therefore what are the ribosomes? Those are the cooks in the school's kitchen. They help to build the proteins. The lunch line is like the Golgi apparatus because it prepares and packages the proteins for use and export. The head custodian in charge of the maintenance crew is going to be like the lysosomes because it's responsible for breaking down and absorbing this unwanted material in the cytoplasm. The support beams, like the walls, the ceilings, and the floors, would be like the cytoskeleton because it maintains the cell shape. And the boiler room of the school is going to be like the mitochondria. It helps to produce the energy. So cell differentiation, what is it? So cells have a complete DNA copy for all of our body functions. Every one of your cells has the exact same copy of DNA in it. But what happens is that we have to turn off the genes that we don't need by those cell type. And so they differentiate and specialize by turning off those genes. This is how we form tissues. All body cells except your sex cells are going to be the sperm or the egg, are going to contain the same 46 chromosomes. What about sex cells? Well, they have to contain half, right? So you get half from your mom, half from your dad. So sperm has half of your dad's DNA, and eggs have half of the mom's DNA. Differentiation is going to depend on which genes are active and which are inactive. So let's just talk about cell division first. So what happens is that first we're actually going to copy that DNA. So cell division is mitosis, and we can abbreviate it as PMAT, prophase, metaphase, anaphase, telophase, PMAT. Okay, so during prophase, this is when the chromosomes are going to condense and become visible. So most of the time what happens is sitting in the nucleus is that we can't really see the DNA. It's not really in a sort of organized fashion as far as we can tell. It obviously is organized, but it looks like a big glob of mess. Okay, but during prophase, they'll actually organize themselves into chromosomes, and we can actually organize those chromosomes on what's called a karyotype, and that's how we can genetically see what the DNA is. The spindle fibers are going to emerge from what are called centrosomes. So centrosomes are over here on the outside, along the poles, so to speak, and we get these fibers that sort of come out, and sort of like Spider-Man shooting out some fibers. The nuclear envelope is going to break down, okay, and the centrosomes are going to move towards those opposite poles. The chromosomes are going to continue to condense, and we're going to start to see some kinetic pores appearing at the centromere. So along the center of each of these chromosomes, or each of the little X shapes, we're going to start to see a kinetochore. The spindle fibers are going to attach to the kinetochores in the center. Then during metaphase, you can see that the chromosomes are all going to line up along the middle during metaphase. That's what meta means. It means middle. Okay? And each sister chromatid is going to be attached to a spindle fiber originating from opposite poles. So this is going to attach over here, and this one's going to attach over here. And so you can see that they're sort of lined up together. Then during anaphase, we're going to actually start to see these sister chromatids pull apart from one another. Okay? So the X's are going to start to pull apart. And certain spindle fibers are going to begin to elongate the cells. Then we have telophase. And telophase is actually when we start to see that nuclear envelope starting to surround those sets of chromosomes. The spindle fibers are going to start to break down. Okay? The chromosomes are going to arrive at the opposite poles. They're going to start to decondense from one another. Okay? And this is where we really start to see them splitting into two separate cells. And then we get cytokinesis. And cytokinesis is when we get in animal cells this cleavage furrow where it separates. Okay. So this is cell division. This is mitosis. And this is how your somatic or your body cells are going to divide in half. So they copy and then they divide. This is meiosis, and meiosis is when we're going to get to our sex gametes, so either the sperm or the egg, right? And so what happens is for males, we get a spermatocyte, which is diploid. Diploid means that it has 46 chromosomes, just like your normal body cell. Okay? We're going to go through meiosis 1, okay? And they're still diploid. But then during meiosis 2, we don't copy again. And so when we divide, we get haploid cells. That means they have half the genetic component. So instead of 46 chromosomes, it has 23. And that's going to give us the sperm cells. And then in females, okay, we get that diploid oocyte. Okay, and we're going to start to, it's already copied, and then it's going to divide. These are genetically the same, but you can see that one of them is going to be a little bit larger. And then we're going to divide again, but not copy, and we get haploid. So again, 23. Now, we don't get 4 eggs. Instead, we get 1 egg and then 3 polar bodies. And polar bodies are going to support that egg cell. You can see that egg cell is much larger than a sperm cell. So let's talk about transport, right? So we have passive transport and we have active transport. Passive transport is where molecules are going to move down what's called their concentration gradient, okay? And so this would be like if you think about people, right? We pile them all into a different space, and when we go through this gate, we start to spread out, right? So we don't want to be all packed in together. This would be like the elevator. When you get onto an elevator with another person, you don't stand directly next to them because that would be weird. So instead, we actually would start to spread out. And molecules are the same. They actually like to have their individual space and they like to spread out. And so when they can start to pass through this selectively permeable membrane, over time, they will start to separate and sort of even out. Okay. So osmosis is this diffusion of water. It's the movement of water molecules. So what happens is that on this side, we have this concentrated sugar solution, and then on the other side, the dilute sugar solution. So not as much sugar molecules. That's what those little blue dots are. They're sugar. Okay. And then in between them, we have what's called a selectively permeable membrane. Now the sugar molecules can't move. And because the sugar molecules can't move, the water is going to move instead. So the water is going to move from this area of low concentration to this area of higher concentration of sugar because it wants to spread out the sugar molecules evenly. Hope that made sense. It's a lot easier to show in a demonstration. So here we go in isotonic median. Isotonic means that it is balanced, okay? And so the water is going to sort of move back and forth as needed. And this is sort of our goal for our cells is an isotonic solution, okay? During a hypertonic median, this would be during periods of dehydration, okay? So we've lost the extra water out here. And because we've lost the extra water out here, our cells are going to move the water out, right? So when we move the water out, our cells start to shrink. Okay? And then this one would be a hypotonic median, meaning that there's not enough solutes on the outside here. So we have some overhydration happening, and the water is going to move into the cell and it could eventually burst. Okay? So what's filtration? Filtration is the movement of molecules, again, due to this pressure gradient, okay? And so we have osmotic pressure. Osmotic pressure is pressure which holds the water. Okay, this would be what's called absorption. Now, remember that inside of our blood, for instance, we have all of these different proteins like albumin, and albumin helps us to hold in that concentration of water within our blood. Okay. And then we have hydrostatic pressure, and hydrostatic pressure, which is pushing against the vessel walls, right? So if I were to turn on a water hose at high pressure, then the water hose would get hard, right? It would be all kind of soft and like really flexible. And then if I turned on the water full blast at the spigot, now it's pushing against the edge a lot, right? And so that's the filtration. So you can see that here, right? So if I turn on the water full blast, it's wanting to push the water out of the tube, okay? Reabsorption is the flow of the water, and it's caused by our blood colloid osmotic pressure. It's from the molecules inside of the blood pulling the water back in. Okay, and so that's the absorption or the reabsorption. Okay, so filtration, the fluid is pushed out of our capillary beds, right? And it does that through our blood hydrostatic pressure. This is the pressure generated by the pumping action of the heart. So every time our heart pumps or beats, right, it's pushing the fluid up against the edges of the vessels. Okay. Reabsorption is by the fluid being pulled back into the capillaries. This is caused by our blood colloid osmotic pressure, and it's created by this concentration of the plasma proteins within the suspension or within the blood itself. We also have other mechanisms for movements, right? We have endocytosis and exocytosis, and this would be going into the cells or out of the cells. So endocytosis on the left, okay? Exocytosis on the right, and it's sort of that opposite, okay? And so what happens is during endocytosis, we're going to sort of pull things into this protective little membrane called a vesicle. And it does that because we're actually going to pull together pieces of the plasma membrane to form that vesicle. And so you can see that what's inside of this vesicle is completely separated from the other stuff inside of the cell. And we want to make sure that it stays separated until we neutralize it. Okay. Same idea here is that we can actually take this vesicle and we can fuse it to the plasma membrane and then send it out. Okay. Membrane potential. So remember, inside of the cells, it's usually -60 to -90 millivolts relative to the outside. We call this our resting membrane potential. Neurons and muscle cells use this membrane potential in order to generate electrical signals by controlling the movement of ions, right? Remember our sodium and our potassium. So remember that inside of the cell, our cell is okay. So if we, hold on one second, I'm going to draw you a little picture. All right, so here's the inside of our cell, right? That's a cell, just pretend. Okay? And inside of our cell, we would say it's okay. So we would draw an O, and then we would put a potassium ion on the inside. Okay, so that cell is okay. On the outside of the cell, we have a lot of sodium. Okay. Now, also inside of the cell, we have some negatively charged proteins. We're going to use a P to show proteins, even though we would normally make it an A, but we're going to make it a P for right now for proteins. So lots of negatively charged proteins. So that means inside of this cell is really negative, and outside of the cell is pretty positive, right, relative here, right? We also have some chloride, okay? We also have some potassium out here. Just like on the inside, we also have some sodium, just not as much. Sorry, it's hard to write. We also have some chlorine in here. Okay, just not as much, right? The big thing here is that potassium on the inside and the sodium on the outside, right? So what happens is when we get a stimulus, okay, let me go back to, let me share this screen. Okay, so here is that inside of the cell, and then there's the outside of the cell. So remember that the concentration of the ions is going to try to maintain homeostasis, but unfortunately the cell can't. And one of the big reasons is because the potassium can actually cross this membrane pretty easily, and the potassium is positively charged, and it's like, hey, I really want to get close to this negatively charged protein here, right? So it's moving over, okay? But the chlorine and the potassium, it can't, okay? So and also that negatively charged protein can't. So what happens then is that we have this sodium-potassium pump. The sodium-potassium pump is going to move 3 sodium out and 2 potassium in using energy. So then we get an action potential, and this is an explosion of electrical activity, and it occurs when a neuron sends a depolarizing current down an axon away from the cell body. Okay, and so remember, we get the stimulus, and it's going to cause the resting potential to move towards zero. So we're sitting down here right at negative 70-ish, okay, and we get a stimulus, and when that stimulus happens, we're going to open up some sodium channels. And then once we hit a certain point of threshold of about negative 55 millivolts, we're going to open up even more sodium channels, right? So the first sodium channels, those are what are called ligand-gated sodium channels, okay? Ligand-gated means that they had to have something bond into them to open. And then we get these more sodium channels opening, and they're voltage gated sodium channels, okay? And they're going to open, and we're going to keep having more and more sodium flooding in. Let me see if I can go back and show you that. Where's my whiteboard? Right? So if I draw you my little demonstration again, right? So we have all of that potassium on the inside, all this sodium on the outside. Sodium is positively charged. The inside of this cell is negative, and I open up a bunch of sodium channels here. Sodium channel, sodium channel. I don't know if you guys can see that little white mark I'm making, but sodium channels, sodium channels. And so when I do that, now the sodium can flood in to the cell, right? And so as more positive is flooding into this negatively charged cell, then we're moving up towards positive, okay, in our graph, okay? Then what's going to happen is we're going to close it all up, okay? So now we have a lot of potassium in here, and we have a lot of sodium in there, okay? And now the inside of my cell is very positively charged, right? I'm up here at the peak. Okay, and so what am I going to do is I'm going to open up some potassium channels, and I'm going to send the potassium out, and the potassium is going to want to leave out. It can already leave out anyway because it can kind of leak anyway out, but now it's like, yes, please let me out of here. I have been in here way too long with this positively charged sodium. Get me out, right? And so I start moving back towards negative. Then I'm going to close up all the channels. And so now my cell has sort of flip-flopped. Now I got a bunch of sodium in here and a bunch of potassium out here. And that is not what I like, okay? And so now, in order to go back to normal, to my resting membrane potential, I got to do my sodium-potassium pump. So I got to get them to flip-flop, okay? All right, so let's go back to the screen. Okay. So we have started here, negative 70 millivolts, right? I'm going to open up my sodium channels due to my stimulus. I'm going to get us up to threshold, negative 55 millivolts. I'm going to open up those voltage-gated sodium channels. And once I hit a particular voltage, which is about positive 30 millivolts, I'm going to close down those channels. Okay? Then my potassium voltage-gated channels are going to open, and all the potassium is going to flood out of the cell. Now my cell is flip-flopped. Okay, now we got to put us all back to normal. So the sodium potassium pump is going to pump us all back to normal. This again is an all or none principle, meaning that if I don't hit threshold, I can't open the rest of these channels. So if I don't hit threshold and open up the rest of these channels, then I'm not going to go. But once I start going, I can't stop it until I do the whole cycle. Okay. Now this is remember exactly sort of that beginning step for skeletal muscle contraction, right? So we get this action potential moving down a neuron. Okay, it's going to release acetylcholine, which is a neurotransmitter, into the synaptic cleft, which is the space between the muscle, the skeletal muscle, and the neuron. Okay, and it's going to cause this action potential. So we're going to see this change of the electrical charge moving through the muscle. And what it does is that it goes down the sarcolemma. The sarcolemma is the cell membrane for a muscle. And it's going to cause the release of calcium because it's going to travel through these two T tubules. And within the T tubules, we have this surrounding it, this sarcoplasmic reticulum, which is just like endoplasmic reticulum, but sarcoplasmic reticulum for muscles. We're going to release out all of this calcium. Okay. The calcium ions are going to bind to troponin, and troponin is going to change the shape. When it does, it removes the blocking action of tropomyosin, and then these actin active sites become exposed. Those actin active sites can then form cross bridges with myosin. It starts to pull the actin filaments towards the center of the sarcomere. It's going to release energy by ATP hydrolysis. And then once the stimulus stops, we're going to have to remove that calcium by active transport. We're going to put it back into the sarcoplasmic reticulum. Tropomyosin blockage is going to be restored. It's going to block those actin active sites. The contraction can end and the muscle fiber can relax. Okay. So neurons need a constant supply of what? Energy or glucose, right? Neurons are amitotic, meaning that they're not doing mitosis. And we need them because they can conduct nerve impulses. They also help to form the blood-brain barrier, which is important to regulate the environment of the central nervous system. So remember that we have cerebrospinal fluid, or CSF, and that's not containing blood, right? So we have to form this blood-brain barrier so that we have CSF close to the brain, and then we have blood away from the brain. Right. So remember, we have our central nervous system. We have the cerebrum, the cerebellum, hypothalamus, the pons, and the medulla oblongata. So make sure that you kind of go through your textbook and review all of those different features and what their main functions are. Here's the cerebrum, the cerebellum. Okay. We have the hypothalamus, which is going to be up in the center-ish. Okay. We have the pons, the brainstem. Okay. Then here we have our autonomic and our somatic nervous system. So remember, autonomic is subconscious control system. So we have the parasympathetic and the sympathetic nervous systems, right? So parasympathetic is the rest and digest. Sympathetic is fight or flight. And then we have the somatic. This is where we voluntarily, consciously control it. So this would be like our muscle movement, our skeletal muscle movement. Okay. Remember, we have our 12 pairs of cranial nerves. Okay. Here's that olfactory for nose. It's a sensory nerve. It's for the sense of smell. Here's the optic, which is for eyes. Okay. Here is our ocular motor for all of our eye muscles, except for those supplied by our 4 and our 6. Okay. The trochlear motor, number 4. Okay. So make sure that you go through these. The trigeminal, which has to do with our facial. Okay. Yeah. Abducens has to do with the external rectus muscles in our eyes. The facial motor is 7. The vestibulocochlear. Okay. There's lots of fun sort of mnemonic devices to remember these in order. Lots of songs, rap songs, etc., that you can find on YouTube. I highly recommend them. There's glossopharyngeal, the vagus, the spinal accessory, and then the hypoglossal with the tongue. So here they are in order: olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, vestibulocochlear, glossopharyngeal, vagus, accessory, and hypoglossal. And also then look at whether it's sensory, motor, or both, and then know the function. Okay. So our autonomic nervous system, we have the sympathetic and the parasympathetic. So remember, in the sympathetic response, that's going to be preparing our body for intense physical activity, so fight or flight. Okay. And so we release norepinephrine at adrenergic receptors. The information is going to be carried via our ganglia and our adrenal medulla. The parasympathetic is going to release acetylcholine at cholinergic receptors. We also have nicotinic and muscarinic receptors. 75% of all of our parasympathetic messages are actually carried by our vagus nerve. Okay. And here is some of our effects of what's happening with our sympathetic or our parasympathetic action. So make sure that you review these. Name all 206 bones in the body and go. Just kidding. Make sure that you review the names of the bones. 106 of them are between our hands and our feet, so make sure that you do review them. These are the muscles that you learned in A&P1, so make sure that you do go through them and that you review them again. Okay. Make sure that you review the nerves. And if you have any questions, you know where to reach me. Please reach out to me at mburke@cecil.edu, and I am happy to help you.